Welcome. Today's lecture is the biochemistry of the immune system. What is going to be explained is characteristic of the immune system, mechanism of the immunity, antigens, non-specific immunity, like phagocytosis, complement, which we already talked about, and then specific immunity like antibodies, B and T lymphocytes, and of course additional information. What is actually going on once the body is infected? Usually there are some responses, like if brain is affected, there is the depression, anxiety, inflammation in lungs, causing cough. If the gastrointestinal tract is affected, there is the constipation, diarrhea, IBD, pain, inflammation in the joints, or in case of breathing pathways, the acne, eczema, rhinitis, and hypersensitiveness are affected. What are the two main mechanisms of immunity, the non-specific and specific? We already talked about non-specific ones, so we are going to repeat it only briefly, and then there are the specific ones. The non-specific immunity is leading to pathogen elimination, and the parts are usually the barriers and so on. On the other hand, the mechanisms of the specific immunity leads to pathogen elimination and moreover immunological memory. What does it mean? That the specific immunity is keeping the memory what already the body encountered and how to fight with it. So the non-specific immunity can be mechanical, like cough, breathing out, coughing, sneezing, and so on. Chemical, which are the proteins and other compounds released by the cells of the non-specific immunity, and microbial, which means usually the phagocytosis. The specific one is somehow different, and we will talk about it later. What is the result is pathogen elimination. What can be the pathogens? Viruses, bacteria, fungus, parasites, and others. Immune system is basic homeostasis maintaining systems, and there is the defense, which is mainly focused on recognition of the foreign. Then there is tolerance, recognition of the body own cells, and immune surveillance, internal harmful substances which are kept down. Immune system is based on anticipation, which means it's standing there and waiting for the thing to happen. Cells and organs of immune system, primarily lymphatic organs like bone marrow and thymus, and secondary lymphatic organs like lymph nodes, spleens, and the parts of the immune systems are cellular components like neutrophils, eosinophils, basophils, mastocytes, monocytes, dendritic cells, B lymphocytes, T lymphocytes, and natural killer cells. Those are the cells and organs of immune system. Cellular part, we already talked about a neutrophil, eosinophil, basophil, monocytes, lymphocytes, and plasmatic cells. So those three we already talked about. Monocytes we mentioned, and the other are going to be subject of today's lecture. Antigen. What is antigen? We have two terms, immunogen and antigen is structure which triggers the immune response, usually is of foreign origin. It is quite often protein or saccharide or lipid, and molecular weight is supposed to be more than 10 kilodalton, usually of complex structure, and there can be hapten, which is small, covalently bound molecule, on bigger structure. So Here we have three uh, different structures of the proteins, and we can see that they have different 3D structure. This is myosin, ovalbumin, and collagen. And what is antigen? Antigen has epitopes which are actually antigenic determinants capable to react with and activate the immune system. They can be sequential and conformational, usually it's both, and there is usually one dominant epitope. It's written here that we can see this is dominant epitope and it can be modified, but still it triggers the immune system. Again, we can see collagen, myosin, ovalbumin, and here what is antigen and epitope. Here we have this primary structure of some of the proteins, and here is something which recognizes the 3D structure of the protein. And here what we can see, this is the special 3D structure. Actually it is presented in simplified form. Then the antibody is capable to interact with the 3D structure but not with the uh, primary structure. So the 3D structure is the thing which is important for the reaction. What is cross-reactivity? Somehow the 3D structures are similar, and therefore the antibody can react even with structures of slightly different proteins, but where the determinant is in the 3D space is really similar. Typical example is birch pollen, which if someone reacts with birch pollen, its immune system, the apple, hazelnut, peach, apricot, and celery can also trigger the immune system. The same applies for latex. It means the natural latex. And there is avocado, banana, kiwi, mites, which are quite common, and allergy to mites is quite common in our countries. And of course the cross-reactivity are crustaceans and mollusks, and wormwood or artemisia, it's carrot celery cross-reaction. Now there are the non-specific mechanisms of defense. First ones are barriers like skin, epithelium, and mucosa. In stomach there is the high concentration of HCl, which means low pH, which is inhibiting a lot of the functions and also destroying or inhibiting a lot of the pathogens. Then there is chemical and cellular defense like phagocytosis and cytotoxic cells, and the reaction of complement. Specific mechanisms are antigen-specific mechanisms, and those reactions are cellular, like T lymphocytes, and humoral-like antibodies. And those actually are keeping the stimulating and keeping the immunological memory. Non-specific versus specific immunity. The innate uh, immunity is first level of defense, is immediate, which means it starts in minutes or hours. For instance, CRP protein starts to be produced in 2 hours, and it's non-specific. It means it follows certain patterns, and it doesn't matter if how similar they are. When they are acquired, which are late and specific, and the target is elimination and immunological memory. Here is the primary and secondary antibody response. If there is the first antibody response, it takes about 7 days for the antibodies to be produced, but once the infection was already over, the second time the body meets the infection, it means repeated, the maximal antibody production is not after 7 or 10 days, but it reaches the peak at about 3 days. And if we take into account that the multiplication of viruses or bacteria are exponential, those 3 days make a huge difference in the infestation. Principles. First is activation of non-specific mechanism, then activation of effector mechanism, and activation of antigen-specific mechanisms. Molecules necessary for immune systems are adhesive molecules which make intercellular contact like integrins, selectins, mucins, then receptors like Fc receptors or complement receptors, and of course membrane proteins like CD markers and enzymes. The other ones are cytokines as a signaling molecules, immunomodulators, and auto, endo, and paracrine effects of those modulators. And there is already one cytokine network. Cytokines are divided into 5 groups. First ones are hemopoietins like interleukin 2, interleukin 3, 6, and so on. Then interferons like interferon alpha, beta, gamma, tissue necrosis factor group like tissue necrosis factor and others, TGF beta, tissue growth factor beta group, and chemokines, which are actually attracting the molecules and sending the signal where they should go. Signaling pathways are YAG-STAT pathways and Toll-like receptors pathway, and usually they are coupled with G protein, phospholipase, and adenyl cyclase. And here we have the basic mechanism of non-specific immunity. Non-specific immunity can be cellular, like high phagocytic activity, granulocytes and macrophages, or humoral, which means complement and acute phase reactants. Recognition mechanism of phagocytes are first PAMP, which are pathogen-associated molecule patterns, which are usually on the surface of endotoxin, proteoglycans on a gram-positive bacteria, glucans, polyglucans, polymannans, and others. And then DMP, damage-associated molecular pattern, and HSP protein. Elimination of microorganism, of course, first one is movement, then adhesion and cell activation, opsonization, phagocytis, and elimination, but some of the steps can be skipped. For instance, we have antimicrobial effectors like myeloperoxidase, lysozyme, lactoferrin, and others we already talked about it. Then proteases like elastase, collagenase, and hydrolysis like cathepsin, phospholipases, and others. What is respiratory burst? We already mentioned it yesterday, and now this is again the structure of the phagocytic molecule, and we can see we have the phagocyte oxidase, and it's producing reactive oxygen species which are actually fighting the bacteria which is already encapsulated in the phagosome, and this is the phagocytic vacuole, and the result is killing of microbes by reactive oxygen species and nitrogen oxide. The high consumption of oxygen is what is called respiratory burst, and usually each molecule can survive up to 5 respiratory bursts. Mast cells, usually they are defense against parasitic infection, usually they interact with allergen-specific immunoglobulin E, And what is important is that once activated, they degranulate and release histamine, serotonin, and other signaling molecules. Here is mast cell and allergic reaction. This is mast cell. Uh, is it has immunoglobulins on the surface, and it interacts with the allergic epitope. Once it is activated, it releases all of the granules and sends the signals further. The high allergic reaction is called anaphylactic shock, and it usually is expressed by weak pulse, hypotension, unconsciousness. In gastrointestinal tract, it's vomiting, stomachache, diarrhea, edema, reddening, rash, itch at the skin and respiratory system, cough, whistling, up to breathlessness and secretion. Those are the allergic reactions, which are actually normal immune reactions, but they react in a higher level and to the things they are not supposed to react. Complement is protein system which is activated in cascade and acquiring enzymatic activity. It's one of the effector during inflammation, and synthesis is mainly in liver. Parts can be synthesized in monocytes and macrophages, and the components are called C1 and C up to C9. There are three ways of component activation. We already mentioned it. So there is classical pathway, alternative pathway, and lectin pathway. And what is the alternative pathway, which is phylogenetically oldest, is that actually C3 part is somehow activated by cutting the C3A part and C3B part. C3B part then interacts with the surface of the pathogen, and then there is the C pathogen with attached C3B part on the surface. Then C3B can convert the C3 to C3A and C3B. It means cut into pieces because C3B is protease, and then it activates other C3B and so on. Then it converts C5 convertase and it leads to lysis. We will talk about it in a while. Classical pathway actually requires the interaction of the pathogen with antibodies, and once the antibody is attached to the pathogen, it is reacting with C1 component, part of the component, which then activates C4 and C2 parts, and then it forms C3 convertases. Against it cuts the C3 part of the component to C3A and C3B, which activate C5 convertase. C5 convertase activates other parts, and what it does is it creates a membrane attack complex. We already talked about it, and the membrane attack complex is somehow like drilling hole in the cell membrane of the pathogen, and it can lead to osmotic cell lysis, or if it is somehow defended, it still which also destroys some of the um, chemical osmotic gradient. Then the other thing is complement C3A and C5A, which are the parts which were released from the C3 and C5 upon the activation. They actually are sending the signal. They are signaling molecules, and the receptors are on the basophil and mast cell surfaces releasing histamines. C3b itself is actually making the opsonization, and the C5b is participating in the osmotics. Like this. Mechanisms of specific immunity, we have cellular components like T and B cells, and antigen presenting a cell, which are dendritic or macrophages or other parts, and then there are the humoral component, which are antibodies. Antibodies usually they are formed from two light chains and two heavy chains, which are uh, attached together by cysteine bond, and usually they interact with a specific 3D epitope, which is here. And once the epitope is activated, it stays there by weak interaction forces. Most of those are hydrogen bonds. Antibodies, they have a high selectivity, they are globular proteins, and here we can see the heavy chains and light chains, which are actually can be divided into smaller parts, and this part at the end is the part which is actually the antigen binding site. We have immunoglobulin G, A, M, D, and E, and usually the immunoglobulin G, E, and D, those are monomers. Immunoglobulin A is dimer, and immunoglobulin M is a pentamer. This is important to remember because immunoglobulin E is bigger and has different, uh, somehow different way how it is interacting. Immunoglobulins, they can be searched by pepsin cleavage, which left the light and heavy chains attached together, and papain cleavage left only the FAB parts, which are only the parts of heavy and light chains, and then there is the FC part, which actually are the remnants of the heavy chains bound together. Polyclonal antibody is antibody which is usually produced by our body, which means if there is some pathogen, we usually don't activate one B cell, but several B cells are activated, and each of the immunoglobulins is actually interacting with different part of the antigen. On the other hand, monoclonal antibody is usually um, antibody of descendants of one B cell, so the antibodies, they have the same 3D structure, they are identical, and they interact exactly with the same epitope. So polyclonal antibodies, they interact with the same antigen but different parts of the antigen, and monoclonal, they interact with exactly the same epitope. Interaction of antigen antibody. So if there is the excess of antibody, the system is soluble, so the antigen antibody is soluble. But if there is the zone of equivalence, there is something which is causing the precipitation, and if the antigen is in excess, again the precipitate is diluted. Here you can see antibody excess, antigen excess, and equivalence is forming the coagulant. It means there is the precipitate form. Here what we can see the interaction of antigen antibody van der Waals attraction, but the most important is hydrogen bond and hydrophobic interactions, which actually choose the specific part of the surface of the pathogen which they bound to. Here is again the hydrogen bond, but in among two water molecules. Affinity and avidity association constant is 10 to 6 to 10 to 12, and interaction usually is one active site. If, which means one epitope. If it interacts with two epitopes, it's called avidity, and it's usually 100 times bigger. And of course, if we have immunoglobulin M, which is pentamer, the avidity can be a lot higher. So avidity is the strength of interaction between the antibody and the epitope, and affinity is usually the strength of the interaction between one of the interaction site of the antibody. B and T lymphocytes. B lymphocytes recognizes only specific antigen determinant, and usually they produce antibodies of the same specificity. The T lymphocyte controls the immune reaction. There are CD4 and CD8-5, and we have TH and TC lymphocyte. It means helper cells and connective cells. Antigen-specific receptors. This is the receptor of a B cell receptor, where we can see that those recognition receptors are actually identical. The antibodies the B cell is capable to produce. On the other side, the T cell receptor has a recognition pattern which is not so actually specially distributed, and there are the CD3 helper sites. And now the presentation of peptide fragments. There is the MHC1 and 2 class, which means main histocompatibility complex 1 and 2 class. Usually they take peptide fragments or produced by the cells or phagocyted by the cells, and they have the CD4 and CD8 binding. So we can see class 1 MHC molecule, cytokines, class 3 molecules, and so on. And those are presented on the surface. Then the major histocompatibility complex takes the microglobulin and it interacts with the CD8 T cell, and the CD8 T cell is actually reacting with the part of the antigen which is presented on the MHC class, and it is actually then activating the other immune system. So first we have the cytoplasmic protein, which is actually making peptides. The peptides are formed by interacting with chaperones, interacting with major histocompatibility complex, and then our Golgi apparatus is presented on the surface, and it's available for the T cells to interact with. Here we can see the major histocompatibility complex 2, and what is important is that it has a slightly different structure, and oligopeptide interaction with MHC and 2 is that major histocompatibility complex 1 is actually presenting smaller parts, and they are enclosed in the MHC presenting part, but the MHC2 presenting part is actually presenting bigger parts, and they are actually slightly more reactive. Cell immunity, we have T helper 1 and T helper 2 cells, and here what we can see is the naive immunoglobulin and cell which interacts with the microbe. Then the B cell is activated with helper T cells and other stimuli, which means the other signaling molecules, then the activated B cell starts to multiply and differentiate, and we can get the immunoglobulin G expressing cells, immunoglobulin N expressing cells, and other cells. And one part is actually forming the memory B cells, which is then stored for uh, for future infection to be easily activated and so on. What does it mean? Usually the stored memory cells are activated, so at the beginning of the infection there are more of the memory cells than before, so the probability of being activated and starting the production of antibodies is higher. Primary and secondary antibody reaction. First is reaction with antigen. B lymphocytes usually stimulates, and resorption is antigen presenting cells. Antibodies of immunoglobulin M, secondary lymphocyte organs, and antibodies immunoglobulin C, isotype transportation. The target is production of memory cells with high affinity immunoglobulins. Those are the primary and secondary antibody reaction. And now there is the mucous immune system, which already was mentioned in the lecture of glycoproteins and proteoglycans. So the mucous system is on the surface of the for of the on the mucosa, and it's preventing some of the pathogen to target the mucous cells itself or the submucous part of the body. Once the mucous immune system can be actually formed by mucous barrier, it's usually epigenetic and environmental. It's the thing which is protecting, and mucous barrier also can interact with immune system, microbiome, and neural system, and all those things together are forming the clinical manifestation. In interaction with nutrition, for instance in gut, there can be antibiotics, alcohols, or dysbiosis, which actually destroy the mucous protective layer, and therefore most of the things which are present in gut, and usually the mucous layer is protected from them, they can go through, and also the antibodies can go through the mucous layer. And once those antibodies are going through, they activate the immune system, and the immune system, once activated, is causing the inflammation, and the inflammation in gut is affecting the gut itself, the circulation, and the storage of the digested parts. So, in summary, immune system is effective defense system. It is the cooperation of innate and adaptive immunity, its ability to synthesize enormous numbers of antibodies, like 10 to 8, and the most important part is the immunological memory, which is the thing which should be uh, one of the main